Hey guys, I wanted to check in with you and kind of tell you what we've been doing here in the lab, so because it kind of goes along with what we're doing there, and I'm probably going to kind of throw some questions about it to some degree. So what we've been doing, we kind of talked about a little bit about Snowmageddon a few weeks ago, when everything was shut down for a few weeks. Somewhere around Thursday, they issued a boil water notice, which they wanted you to boil their water because it could have had pathogens in it. On one of the previous videos, I showed you that there's not supposed to be any coliforms in drinking water, and so they're worried when pressure falls that in the system that there could be a crack in the pipe, which then pathogens from the soil could enter the pipe and then get to your house and then make you sick. So they tell you to boil the water because that'll kill most of the pathogens. So what we've been doing here at school is kind of the test that they've been doing. So again, Thursday they put out the boil water notice, and then Saturday they cleared us from the boil water notice. And the reason that it took so long is because this is basically the process that they use. So this is a coliform test vial, and this solution that's in here, if coliforms are present, it'll change color. So what we did here at the school is we went around and we tested some different locations. So like this was location 1, which happened to be a toilet, and after 24 hours you go in and you check the color, and if the color is different, this one has not been used, and then this one has 1 milliliter of water from a toilet here on campus added to it. So notice that color wise, toilet water is clear. You could drink it. It's ready to roll. There are no pathogens in the toilet here, and that's because municipalities in the United States use drinking water for toilet water. So we'll talk more about that maybe later. I've already beat people down here at school about how dumb it is to treat water, release it to the lake, then pull it back in, treat it, send it to your house. But anyway, so again, we checked a bunch of places, and a lot of places ended up pretty good. And then to kind of give us a control, we know that water outside interacts with animals all the time, and there tends to be fecal coliforms out there. So this one turned yellow, and so when bacteria are present, they create, just like we do. They have to breathe in and out and give off different chemicals and whatnot as part of their metabolic process. And so the chemicals that they give off mix with this solution that was in here, and it'll turn it yellow if they're there. And so a couple things happened when we did this. I did not plan it this way. Usually it doesn't happen, but occasionally it does happen. And what happened with ours, let me see if I can find it. I missed it, so hold that thought. I gotta look for something. Sorry, found it. It was right here on my desk. Anyway, every once in a while what happens is this guy here and that guy, notice that there is a little bit of cloudiness in this, and it does kind of have sort of a little bit of a yellow tone to it, and this was a bottle dispensing filling station. Like here on campus, if you haven't been here for a while, they've got a few bottle filler water fountains now, so it's got a place to sensor fills your bottle, and this is what one of them struck as, which scared us a little bit, because you don't want to be drinking from that. So what we did the next day, just to make sure, this was after 24 hours, it does have a little bit of a color change, and you'll be able to see it here, but it's very subtle. It's not crazy, but there is a difference in these two chemicals or colors. So let me kind of do that. You can kind of tell if I do that. So this one on the let me do it this way. This one is the one that came back as questionable, and then what we decided to do is make sure that it wasn't just user error on the first one, contamination. So what we did was we went back to that same bottle filler the next day after we had this kind of positive test, right? And we got another sample were a little bit more careful that we didn't do any kind of cross-contamination, and after 24 hours we got this. But basically what you're supposed to do is check these at 24 hours. If there is no color change, then we need to wait another 24 hours, because sometimes it can take up to 48 hours for this to show itself. And if it does show up yellow, then that indicates that there are pathogens present. If it doesn't show up yellow and it stays that red color or whatever, then you would lift the boil notice, which is basically why they shut the water down on Thursday and then reopened it on Saturday, because they did this very test, which takes 48 hours. So Thursday, they checked it again Friday, and then on Saturday, they checked it again and it was good. So they said, go ahead and start drinking your water. So what the other test that they were doing while that's going on is, so that you don't lose a lot of days, when they tested those samples by putting some in a vial, they also put some on a count plate, which initially will look like this, and the little red stuff inside of that is nutrient agar, which is just food for bacteria. And if after 24 hours of incubation, these are bacterial colonies, or fecal coliforms, or coliforms really. We don't know if they're fecal coliforms for sure, but when they do show up on here, they are definitely coliforms. And on these plates, there's actually a way to count how many are on there, but those navy blue colonies are more than likely the kind of nasty bacteria that we're looking for that could make humans sick. So this is the kind that typically would be found in mammalian or human feces, so it might be E. coli. So what we're doing with this, first of all, you can get a count from that. This one I think we counted at 39 coliforms per milliliter, which, if we're looking at our limitations from that video before, 3,900 per 100 milliliters would be what this looks like, and 3,900 would be in the realm of fish and wildlife habitat. You wouldn't want to drink it. You would, obviously, right? You wouldn't want to drink it. You wouldn't want to recreate in it. So you don't want to go swim in the area out in front of the school, because that's where this sample came from. So basically, with this, what we then do is we take these colonies and we can isolate them onto Petri plates, which I'll show you the result of that in another video, because those are waiting. So today we went in and we took a smidgen of these colonies. Looks yummy, right? And we're trying to isolate them on another plate. And then next time we're going to come in and look at them underneath the microscope to see what they look like. If we know what they look like, then that would kind of give us where tests would go next. So basically what we've been doing, day before yesterday we got our first water sample. Today we got the official findings of if it's good or bad, if you can drink it or not. Most of them are drinkable, including the toilets. Today we also isolated the ones that do have bacteria in them, and we are going to further test those so that when we come back in, really it would be if we came back in Saturday, we could test them, but we're not going to be here Saturday, so on Monday we'll come back in and we'll test them. So really we're looking at about a 5 to 6 day span before we can just get through the first phase of identifying what type of bacteria is in here. And then it might take us anywhere from a week to a month to isolate these even further and figure out exactly what species of bacteria they are. So sometimes people wonder why it takes so long. It's a long process. You have to do a lot of time in between waiting. And then a lot of people wonder why we don't test water more frequently. Well, it's expensive, time consuming. And then it also takes a lot of manpower to do all of that if you're continually doing it. So that's the lab that we're working on now. Again, I'll show you another video later that'll kind of break that down for you. So until then, enjoy this one.